There are three main types of genetic testing: gene testing, chromosome testing, and biochemical testing. Let's explore. Gene tests are used to look for signs of a disease by examining DNA taken from a person's blood, body fluids, or tissues. Let's take a journey into our cells. The test can look for large changes, such as a gene that has a section missing or added, or small changes, such as an altered chemical base with a DNA strand, genes with too many copies, genes that are too active, genes that are turned off, or those that are lost entirely. One technique for genetic testing is called DNA sequencing.
Chromosome tests involve the analysis of chromosomes, as various genetic syndromes are caused by structural chromosome abnormalities. To analyze a person's chromosomes, the individual cells are allowed to grow and multiply in a laboratory until they grow to a certain stage. Under a microscope, the chromosomes are counted, identified, and analyzed based on their size, shape, and stained appearance. Genetic diseases are associated with changes in chromosomes such as extra or missing complete or portions of chromosomes, or exchanges of materials known as translocations between chromosomes. Chromosome tests can be done two different ways. In a karyotype test, the chromosomes are cut out and arranged side by side with their partner chromosome in ascending numerical order, from largest to smallest. The FISH test identifies small pieces of chromosomes that are missing or have extra copies, possibly missed by the karyotype test.
Biochemical tests look at the amount and level of key proteins. These levels can identify genes that are not working normally. Newborn screening uses these types of tests, for example, to detect infants who have metabolic conditions such as PKU. There are currently many applications of genetic testing. Prenatal screening is when the genes of an unborn child are tested to see if they have any problems or disorders in their genome. Prenatal testing is done on the fetal cells by chronic villi sampling from the baby's developing placenta. These cells grow faster and can be analyzed sooner than if they were taken using a different method. Prenatal screening tests for Tay-Sachs disease, cystic fibrosis, Down syndrome, Turner syndrome, sickle cell disease, spina bifida, and many others. Different religious beliefs have very diverse opinions on this subject of screening.
Scientists remove cells from embryos in vitro, test them, and use results to determine the embryo's characteristics or the nature of the embryo. Gender selection is usually a big reason for couples to choose pre-embryo testing. There are many controversies about this subject as well. Dear Diary, I've been thinking a lot about pre-embryo testing, but I'm not sure if it's ethical. I mean, I really want a boy. Craig and I have always wanted a boy, and we already have 2 girls, so I mean, it wouldn't make sense to have another girl. But pre-embryo testing doesn't seem right, and it's expensive.
Dr. Martone is with us today, and she's going to be talking to us about carrier testing.
Thank you, Meredith. Carrier testing is used for individuals possessing a gene associated with a disease but never exhibit any symptoms. A carrier is a person who is not affected by the mutated gene he or she possesses, but risks passing it on to future generations. If one or both parents are a carrier, the risk of passing on the disease to their offspring is likely. To predict this risk, it is necessary to know if the gene is autosomal or X-linked. This type of testing is offered to people with genetic disorders in their family history or people of a certain ethnic group.
There has been a new breakthrough in genetic testing. Pharmacogenic testing examines a person's genes to gain information about how drugs will be broken down by the body.
Pharmacogenic testing aims to design drug treatments that are specific to each person and help them obtain the right amount of medication.
Dr. Martone, could you please explain to our viewers what diagnostic testing is?
Sure, Meredith. Diagnostic testing is used to confirm a diagnosis when a person has signs or symptoms that suggest a genetic disease. It can be performed at any time in a person's life, including before birth. This particular genetic test depends on the disease for which the person is being tested.
The testing of hundreds or even thousands of genetic variations at a time from a DNA sample is called multiplex testing. The variations are studied, then eventually diagnosed. Some of the variations would most likely contribute to a genetic disease. This testing is very expensive and is considered unethical.
So can you explain to me exactly what you're doing?
Well, newborn screening is testing just after birth to identify disorders that can be treated early on in the child's life to control his or her disorder.
What kind of disorders are you testing for?
We test for a wide range of disorders.
How is the newborn tested?
The newborn screening tests infants' blood samples for abnormal or missing gene products. I've always wondered what pre-symptomatic testing is. Dr. Martone, could you please enlighten me?
Pre-symptomatic testing is used to detect late-onset genetic disorders that do not show their effect immediately. If a speculated mutated gene is responsible for a late-onset disease, and it has been identified, a person from the affected family can be tested before symptoms appear. The validity of looking for genes in a person without symptoms or without family history of the disease is currently still being researched.
Thank you so much, Dr. Martone, for coming in today.
It's my pleasure, Meredith.
Hi everyone, my name is Meredith, and I've actually just heard about this really cool thing on the internet. It's a genetic screening test that you can do at home, so I'm going to try it out. So it says here that all I have to do is scrape a couple cells off the inside of my cheek. It doesn't sound that bad.
Why did you choose this instead of going into a clinic to be screened?
Well, it's a lot more accessible, and I don't have to wait for an appointment, and I feel like my results might be more private, so just to me rather than to my doctor in the hospital.
Right. Wouldn't there be any risks?
No risks. The only difference is that there's a lack of government regulation, and the potential misinterpretation of genetic information is greater.
Not bad, not bad.
Finally got it in the mail, so I'm gonna try it out.
That was easy.
That was easy.
So how exactly do you get the results?
I get it in about a month, and they'll email it to me. So it's been a month, and I finally got my results, so I'm gonna check them out. My genes are clean!